So this is the first day of our 2024 A-level English medium biology paper class. Now we faced for a placement test, right? So you don't like only two children answer this paper. It's okay. Others can download the paper from the Telegram channel and answer the paper later, right? So don't worry about the marks of this paper. This is just to know what is your current level. දැන් ඔයගොල්ලෝ ඉන්නේ මොන level එකේද කියලා බලන්න විතරයි. And this is from grade 12 syllabus. Right? Yes, let's start the discussion with the MCQ paper. හරිද? Now here I'm going to discuss all the questions, right? One by one. Now, if we take the MCQ paper, you know there are 5 choices for each one. නේ? එක ප්‍රශ්නයකට answers 5ක් තියෙනවා. එතකොට අපි biology MCQ එකක් discuss කරනවා නැත්නම් කරනවා කියන එකෙන් our only target is not to find the correct answer. හරි? paper එකක් practice කරන කාලෙදී අපේ aim එක වෙන්න ඕනි, target එක වෙන්න ඕනි ප්‍රශ්නෙට හරි උත්තරේ හොයන්න විතරක් නෙමෙයි, right? The other thing is, the most important thing is, we should know why the rest of the other 4 answers are incorrect. ඇයි අනිත් උත්තර හතර වැරදි කියලා තමයි අපි හොයන්න ඕනේ. තේරුණාද? That is how you have to practice MCQ papers. Okay? Right. දැන් මේක කරද්දී ඔයගලන්ට පුළුවන් මොකක් හරි වෙනම book එකක් එහෙම නැත්නම් වෙනම sheets ටිකක් අරගෙන කියන ඒවා ලියා ගන්න. හරිද? එතකොට now here we have only 30 MCQs. But in every MCQ, they have 5 choices. So at the end of this discussion, you are going to learn, right, 30 into 5, new 150 points. Points 150ක් අපි ඉගෙන ගන්නවා. හරිද? Right. Now, let's start the discussion. Number 1. In the order and organizational level of living matter, in which level plant can be replaced? Now, you know there's a hierarchical order. Right? So, now if we take a plant leaf, it is made up of different types of tissues. Tissues, they are group of cells. So, we can identify plant leaf as an organ. Tissues, ජාති කිහිපයක් එකට එකතු වෙලා තමයි මේක හැදිලා තියෙන්නේ නේ. Now, if we take a dicot leaf, there are palisade, spongy bundle sheet cells, mesophyll cells, right? ඔය විදිහේ එක cells තියෙනවා. Right? Cells එකතු වෙලා tissues හදනවා. A tissues, one or more type of tissues, they get together and form organ. හරිද? So, for the first question, the answer should be second answer, organ. Then, question number 2. Contains only branched polysaccharide. Now, this is from unit 2, right? Carbohydrates. Carbohydrates වලනේ අපි polysaccharide එකක් ගැන කතා කරන්නේ නේද? You know, carbohydrates we can classify it as monosaccharide, disaccharide, and polysaccharide. There's another group called oligosaccharide, but that is not required for your A-level syllabus. Polysaccharide කියන්නේ මොනවද? One or two or more subunits, right? Now, if we take monosaccharide, ඒක කියන්නේ monomers. එයා දැන් අපි ගත්තොත් glucose එක එතන එයා තමයි monomer එක. එතන එක්කෙනයි ඉන්නේ. එතකොට ඒ වගේ දෙන්නෙක් එකතු වෙලා මොනවද හදන්නේ? Disaccharides. එතකොට monomers ගොඩක් එකතු වෙලා මොනවද හදන්නේ? Polysaccharides. They form polysaccharides. Now, based on the architecture of this polysaccharide, we can classify or we can categorize polysaccharides as linear and branched, right? Now, in the syllabus, they have given some examples for linear form. Cellulose, glycogen, inulin, right? And branched form, glycogen is branched form. හරිද? ඒක linear නෙමෙයි. Amylopectin and hemicellulose. Linear එකට එනවා amylose එක. Right? So now here we have to find the answer which contain only branched polysaccharide. එහෙනම් කොයි එකේද තියෙන්නේ අපිට? එහෙම උත්තරයක් හම්බෙනවා third one. Hemicellulose, amylopectin and glycogen. Now let's talk about let's discuss about the functions of these polysaccharides. අපි එකින් එක ගමු. Right. First one, අපිට ගන්න පුළුවන් amylose. Right? amylose ගත්තහම starch නේ, starch වල තමයි forms දෙකක් එන්නේ, amylose and amylopectin. එතකොට amylose ගත්තහම එයාගේ මොකද තියෙන්නේ? linear. එයා linear polysaccharide එකක්. But amylopectin, it is a branched. Anyway, starch, you know, it is a storage polysaccharide. Anyway, but right, the function is storage, right? So starch, they're stored in plants and green algae. Next one, glycogen. In glycogen, what is the monomer? Glucose, right? It is a branched polysaccharide. And also the function is storage, right? Stored in animals and fungi. Now, if we take vertebrates, glycogen is stored in liver and muscle cells. Cellulose, again, the monomer is glucose, but this is a linear and structural polysaccharide, right? So the function is, it is a component of cell wall of plants and algae. Next one, inulin, right? Inulin, the monomer is fructose. Monomer එක වෙන්නේ දැන් fructose එකක්. This is a linear polysaccharide and the function is like it shows storage functions. Stored in tubules of dahlia. Next one, we have pectin. The මෙතන නැහැ pectin කියන එක න. Anyway, pectin is also a polysaccharide. The monomer is galacturonic acid. This is a branched and structural polysaccharide. It's a component of middle lamella of plant cell walls, right? Then hemicellulose. Then the monomer pentoses. Pentoses තමයි මෙයාගේ monomers ලා ඉන්නේ. Right? This is a component of plant cell wall. Next one, chitin. Chitin is a nitrogen containing polysaccharide. Now, if you know carbohydrate, අපි කියන්නේ කොහොමද definition එකේ? Carbon තියෙනවා, hydrogen තියෙනවා, oxygen තියෙනවා. හැබැයි මේ chitin ගත්තාම, right, it has a nitrogen group. OH minus group එකක් replace කරලා ඒ තැනකට nitrogen bond වෙලා තියෙනවා. Right? Chitin. The monomer is glucosamine. This is a structural polysaccharide. Right? It's a component of fungal cell wall and exoskeleton of arthropod. Now, anyway, here in this question, they're asking about the branched polysaccharide. So the answer should be third answer, hemicellulose, amylopectin and glycogen. Right, look at the third question, that is from microscope. Right, microscope වලින් තමයි ඒක අහලා තියෙන්නේ. Which of the following statement is correct regarding microscope? Right, now if we take microscope, there are several categories, right? Light microscope, දැන් අපි ගත්තාම අපේ hand lens. It is a simple light microscope. ඊළඟට අපේ lab එකේ තියෙනවා මොකද්ද? Compound microscope. එතනින් එහාට ගියාම තව තියෙනවා electron microscope. Right? Anyway, what is the principle here? මොකද්ද මෙතන principle එක? Right? Visible light, it passes through the specimen and then through the glass lens and produce a magnified image of that specimen. Specimen එක magnify කරනවා. ඕක තමයි principle එක උතන වෙන්නේ. Right? Now we have to find the correct statement. ඒ කියන්නේ හතරක් වැරදි. බලමු අය හතරක් වැරදෙන්නේ. In light microscope, visible light first penetrate the objective and then through the specimen. Now you know in the compound microscope there are two lenses. ඒ කියන්නේ compound කියන්නේ හරිද? එකක් තමයි eyepiece එක, which is closer to the eye, and the other one is the objective lens, which is closer to the specimen. Right? In light microscope, visible light first penetrate the objective. No. Right? What happened here is light from the object or the specimen on the slide, right, passes first through the objective lens and it produces a magnified image, right? Then that image acts as an object for the second lens. What is the second lens? Eyepiece, and magnify that image further. Okay, now the method කියන්නේ visible light first penetrate the objective and then through the specimen. Specimen එක තමයි ඉස්සෙල්ලාම visible light එක specimen එකේ, ඊට පස්සේ objective lens එකට යනවා. හරිද? නැත්නම් objective එකෙන් specimen එකට යනවා නෙමෙයි. So the first statement is incorrect. ඔන්න අපි එක point එකක් ඉගෙන ගත්තා. ඊළඟට කියනවා in the electron microscope light rays are projected through a vacuum. Right? Yes, vacuum එකක් තමයි use කරන්නේ. That's why we can't use not living specimen. You know, in the electron microscope, we can't use living specimen. Living specimen එකක් observe කරන්න බෑ. දැන් අර grade 10 වලදී එහෙමනම් ඔයගොල්ලෝ බලලා ඇති පොණ්ඩ එකේ ඉන්න මාළුවෙක් අල්ලන් ගිහිල්ලා සාරිගප්පියෙක්නේ අල්ලන් ගිහිල්ලා මොකටද කරන්නේ මයික්‍රොස්කෝප් එකේ දාලා ඔයගොල්ලෝ බලනවා ඔගේ අර blood circulatory system එක. තුව මරලා නෙමෙයිනේ බලන්නේ. It's a living specimen. But in the electron microscope, we can't use living specimen, right? Only non-living specimen can be observed. Because why? Then a vacuum එක තමයි අපි තියන්නේ අපේ specimen එක. Right? That is okay. But light rays are projected. No, light rays නෙමෙයිනේ. දැන් මේ light rays වලින් තමයි මේ ප්‍රශ්නේ ඇති උනේ. ඒ හින්දානේ electron microscope එකක් develop කරේ දන්නව නේද? Now, you know the resolution power, right? It is limited by the wavelength, right? Resolution power, it is inversely proportional to the wavelength. Yani wavelength එක වැඩි වෙන්න මොකද වෙන්නේ? Resolution power එක අඩු වෙනවා. හරිද? අන්න ඒකට solution එකක් විදිහට තමයි electron microscope එක develop වුනේ. So in electron microscopy, a beam of electrons, not light rays, a beam of electron is focused through the specimen or onto its surface, right? So that is why the second statement is incorrect. Harida. Next one, third one, scanning electron microscope is used to study the detailed structures of the cell. Scanning electron microscope. Now in our syllabus we have two types. There are two types of electron microscopes: transition electron microscope and scanning electron microscope. In the transition electron microscope, TEM right? It is used to study the internal structures of the cell. Internal structures, right? Now what happened here is a beam of electron passed through a thin, specially prepared slice of material, right? Then a very thin specimen is used here. Harada. Now, this transition electron microscope, it produces 2D image and it has a high resolution. So this is used to study the internal structures, right? And the scanning electron microscope, right? Here in the scanning electron microscope, it is ideal to observe the surface view, right? 3D surface view එකක් ගන්න තමයි scanning electron microscope එකක් ගන්න. කියන්නේ පිටත පෙනුම. එහෙනම් එතනදී අපිට detail structure එකක් ගැන study කරන්න පුළුවන්ද? බෑ. Right? So to study the detailed structure of cells, we have to use the TEM, we have to use the other one, right? Transmission electron microscope. So third statement is also incorrect. Look at the fifth one. Transition electron microscope is used to study the detailed structure of living specimen. දැන් මම කලින් කිව්වා අපි ආව detailed structure එකක් බලන්න ගන්න පුළුවන්. ඒක වැරදි. ඇයි? Living specimen. Living ඒවා බෑ. නේ? Electron microscopes are used to observe non-living specimens. So the fifth statement is also incorrect. Now only fourth statement is remaining. Let's see. Magnification and resolving power are the important parameters of the microscope. Yes. Right? What is magnification? Magnification is the ratio of an object image size to its actual size. And resolution power, it is the minimum distance between 2 points that can be distinguished as 2 separate points. So magnification and resolving power are the 2 parameters of a microscope. For the third question, answer should be fourth answer. Next one, fourth question. What is true about benign tumors? Right? දැන් ඒක අර syllabus එකට අලුතින් add වෙච්ච කෑල්ල. Tumors, goals and cancers, right? ඒක තියෙන්නේ කොහෙද? අර cell division වලට පස්සේ තියෙන්නේ. Tumors and cancers. Normally, you know, this cell division, it is cell division, it is driven by external or internal factors, right? But now if we take cancer cells, they don't respond to the body control mechanism. Cancer cells a body control mechanism එකට respond කරන්නේ නැහැ. ඒගොල්ලෝ හිතේ හැටියට divide වෙනවා. Divide වෙලා මොකද කරන්නේ? ඇඟ පුරාම තියෙන ඔක්කොම organs ඒවා attack කරනවා. Right? They divide excessively and they invade other tissues. Check කරලා අපි detect කරගත්තේ නැත්නම් ඇඟ ඇතුලේ cancer එකක් තියෙනවා කියලා මොකද වෙන්නේ? ඒක මුළු ඇඟේම පැතිරිලා ගිහින් that cancer can kill that organism. Right? මිනිස්සෙක් ගත්තොත් මැරෙන්න පුළුවන්. හරිද? Now here what happened is then ඔන්න කොහේ හරි ඇඟ ඇතුලේ cancer එකක් ඔය විදිහට cells body cells ඒවා වෙනස් වෙලා cancer එකක් abnormal cells produced and what happened that එක single cell එකක් තමයි ඉස්සෙල්ලාම ඔය විදිහට abnormal වෙන්නේ කියලා ගත්තොත් right they undergo then transformation right transformation means a normal cell converting into an abnormal cell හොඳට තිබ්බ cell එක දැන් abnormal වෙනවා. ඔකට කියනවා transformation. Then අපේ body immune system, if the body immune system cannot recognize that and destroy, what happens? It leads to proliferation of cells, right? And it forms a tumor. තව cells divide වෙලා මොකද වෙන්නේ? Tumor එකක් හදනවා. හරිද? මෙන්න මේ tumor එක, right? මේ cells ටික තියෙන original site එකේම ඉතිරි වෙලා තියෙනවා නම් ඒ lump එකට අපි කියනවා මොකද්ද? Benign tumor කියලා. හරිද? Benign tumor it is not a serious problem and it is not considered as a cancer because it can be removed by a surgery. The problem is the malignant tumor. Malignant tumor means they can attack one or more organs of the body. හරිද? malignant tumor එකක් තියෙන කෙනෙක්ට අපි කියනවා අයක් cancer එකක් තියෙනවා කියලා. right? now benign tumor එක ගැන තමයි මේ ප්‍රශ්නය අහන්නේ. Spread distance from their original site. No. ඒගොල්ලන්ගේ original site එකේම ඉන්නවා. Spread වෙන්නේ නැහැ. Become invasive and attacks one or more organs, that is malignant tumor, right? Not benign. Can be completely removed by a surgery, yes, that is correct. Multiplication can create a new tumor? No. Attacks one or more organs? No. Right? So for the fourth question, the answer should be third answer. Next one, fifth one, that is regarding cell junctions. Select the correct statement regarding cell junctions. Now, you know, for the 2024 A-levels, you have to use the new resource book. Then දෙදස් 23ට අර අලුත් එකයි පරණ එකයි දෙකම use කරන්න ඒගොල්ලෝ accept කරානේ. දැන් ඔයගොල්ලන්ට තියෙන්නේ අලුත් පොත විතරයි. හරිද? එතකොට දැන් පරණ එකට වඩා අලුත් එකේ දැන් පරණ එකේ cell junctions වල තියෙන definitions කිහිපයක් අලුත් එකේ අයින් කරලා තියෙනවා. හරිද? right. බලමු. ඉස්සෙල්ලාම cell junction එකක් කියන්නේ කොහොමද? Cell junction means cell junctions කියන්නේ the structures at which neighboring plasma membranes are joined, right? And they can interact and communicate via direct physical contact. Now here we have to select the correct statement. Tight junction prevent leakage of substances, right? Then fifth that is the correct answer, right? Tight junctions prevent leakages of substances. Yeah, that is correct. බලමු second one එකයි වැරදි කියලා. Cell wall of adjoining cells are joined in cell junction, not the cell wall, plasma membrane. Plasma membrane එකයි join වෙලා තියෙන්නේ. So the second statement is incorrect. Third one, plasmodesmata found in some animal cells. No, plasmodesmata they found in plant cells only. Plant cells වල තමයි plasmodesmata තියෙන්නේ. Right? Plasmodesmata are microscopic channels that run through the plant cell wall. හරිද? ඒගොල්ලෝ නිකන් අර අපේ animal cells ලගේ cells වල තියෙන gap junction එක වගේ. Right? But found only in plant cells. Anchor junction වල exchange of materials. No, that is done by gap junctions. Right? Anchor junctions, they mechanically attach the cytoskeleton of adjoining cells by intermediate filaments, right, for strong binding, right? They don't involve in exchange of materials. So fourth statement is also incorrect. Fifth one, communicating junctions found between skin epithelium. Communicating junctions, gap junctions, right? They provide cytoplasmic channels from one cell to an adjacent cell. And these gap junctions, they consist of special membrane of proteins that surround the pore through which these iron, sugars, amino acids pass, right? And found in the heart muscle and animal embryo, right? So we can't find the communication junctions not found in the skin epithelial cells. So that is also incorrect. Answer should be first answer. Next question, sixth one. Select the correct statement regarding enzymes. Enzymes වලින් අහලා තියෙනවා. What is the definition of enzymes? Enzymes කියන්නේ මොනවද? Enzyme, it is a macromolecule, right? Which act as a biological catalyst. And they produced only in living cells. Produced, right? Then next one, the optimum temperature for enzyme activation of bacteria in hot spring is around 50 degrees Celsius. No, that is 70 degrees Celsius. So that statement is also incorrect. Next one, ATP acts as an allosteric inhibitor and increase the catabolic rate. මෙතන මොකද්ද? ATP නෙමෙයි. ADP නේ. ADP function as allosteric activator. ඕක අපි කොහෙද ඉගෙන ගන්නෙ? අපි ඉගෙන ගන්නවා allosteric activation and inhibition කියන part එකේදී. Right? මේ ගැන ඉගෙන ගන්න. Here ADP function, not ATP. ADP function as an allosteric activator and bind to the enzyme and stimulate the production of ATP by catabolism. Right? When the ATP supply exceeds the demand එක exceed කරාම මොකද වෙන්නේ? ATP bind to the same enzyme as an inhibitor. Right? As an inhibitor and decrease the catabolism. So the third statement is incorrect. එතන කියන්නේ මොකද්ද? Increase the catabolic rate. Increase කරන්නේ නැහැ. Decrease කරන්න. Fifth one, the optimum pH for trypsin is 2. No, the optimum pH for trypsin is 8. So the answer is fourth answer. Most allosteric enzymes are made up of 2 or more subunits. That is the correct answer. Seventh question. Correct statement regarding non-vascular plants. Right? Non-vascular ගත්තහම අපිට තියෙනවා පුගුණාටම් මෙහෙම නේද? There are some sporophytes completely depend on gametophytes. That is correct. Okay? Sporophytes are bisexual. No. Some club mosses show heterospory. That is also incorrect. All are contains of tracheids for water conduction. There is only one female gametophyte developing to megasporangia, means spike mosses. Now here the answer should be seventh answer. For the seventh question, answer should be first answer. Here the haploid gametophyte is the dominant stage in non-vascular plants, right? And the sporophyte is not independent. It is completely dependent on the gametophyte. Question number 8. The main reason for the low production of oxygen in bundle sheet cells is bundle sheet cells වල oxygen අඩුවෙන් හැදෙන්නේ. ඇයි? ඒකේ pH2. Photosystem 2 එක it is depleted. Right? Then අපි අර blight reactions ගත්තාම splitting of water takes place in the photosystem 2. Thylakoid membrane එකේ photosystems ජාති දෙකක් තියෙනවා. PS1 and PS2. PS2 කියන්නේ P680 chlorophyll A molecule එක තියෙන එක්කෙනා. හරිද? එතකොට එතන තමයි splitting of water takes place. Right? Oxygen is produced by splitting of water. එහෙනම් PS2 නෑ කියන්නේ මොකද්ද වෙන්නේ? එතන no splitting of water, so no oxygen production. Right? So the reason for the low production of oxygen in bundle sheet cell is decreasing PS2, photosystem 2. Answer number 3. Question number 9. A feature that can be seen only on two specific domains. Domains අපේ කීයද? 3යි. Bacteria, archaea and eukarya. හරිද? එතකොට domains දෙකක විතරක් තියෙනවා. බලමු. Formyl methionine is the initiator amino acid for protein synthesis. Protein synthesis වල අපි ගත්තාම formyl methionine එහෙම නැත්නම් methionine. මේ formyl methionine found only in domain bacteria. හරිද? Eukarya and archaea they have methionine. So that is incorrect. Then there is only one type of RNA polymerase. දැන් අපි ගත්තාම domain bacteria they have only one kind. Whereas archaea and eukarya they have several kinds. Then antibiotics inhibit growth. Now antibiotics, they can streptomycin, chloramphenicol, right? They wage antibiotics, they can inhibit the growth of bacteria, right? Organisms belong to domain bacteria. But domain archaea and domain eukarya, their growth is not inhibited by antibiotics. Many genes have introns, right? Introns කියන්නේ මොනවද කියලා you will learn in unit 7 under molecular biology. හරිද? එතකොට මේ introns අර ඔයගොල්ලන්ගේ තියෙනවා වෙනම table එකක් unit 3 එකේ එතන දීලා තියෙනවා. Right? Introns in genes very rare in bacteria. Right? In archaea present in some genes, but in eukarya present in many genes. Last one, presence of histone proteins associated with DNA. Right? In bacteria, domain bacteria, histones associated with DNA is absent. Right? No histone. But in archaea and eukarya, they have histone proteins. හරිද? දැන් මෙතන බැක්ටීරියාට histone protein එක නැති වුණාට there are some other associated proteins. වෙන proteins තියෙනවා. histone තමයි නැත්තේ. හරිද? ඒක මතක තියාගන්න. So for the 9th question, answer should be 5th answer. Question number 10. Sinusoidal fungal filaments can be seen in. Sinusoidal කියන්නේ මොකද්ද? Sinusoidal means they lack septa. Septa කියන එක නැහැ. මොකද්ද මේ septa කිව්වේ? මෙන්න මේ විදිහේ. Then fungal filaments ගත්තාම මේ තියෙන fungal filament එකක් කියලා හිතන්නකෝ. Right? This is a sinusoidal fungi. Right? මොකද මේක කොහෙවත් අර compartments වලට වෙන් වෙලා නැහැ. septa කියන જાතිය නැහැ. මේ විදිහට ඔක්කොම නියුක්ලියස් ඔක්කොම තියෙනවා එක. එක සෙල් එකක් විදිහට තමයි තියෙන්නේ සම්පූර්ණ ෆිලමන්ට් එක. එකක් විදිහට. හැබැයි මේ septate කියන්නේ මේ ෆිලමන්ට් එක compartments වලට වෙන් වෙලා තියෙනවා. right? මෙන්න මේ විදිහට මේක බෙදිලා තියෙනවා. right? එතකොට එක compartment එකක් ඇතුලේ එක nucleus එකයි. ඒ විදිහට. right? And at the middle of the septa they have a opening. Opening එකක් තියෙනවා. ඒ opening එක හරහා අර තියෙන organelles වලට එහෙම යන්න පුළුවන්. හරිද? Sinusoidal means they lack septa. ඒගොල්ලන්ට septa කියන එක නෑ. හරිද? එතකොට සෙප්ටා නැත්තේ Chytridiomycota and Zygomycota. Right? Chytridiomycota and Zygomycota, they are sinusoidal fungal filaments. ඒ කියන්නේ they lack septa. Chytridiomycota examples Chytridium and Allomyces. Zygomycota examples මොනවද අපි ඉගෙන ගන්නේ? Mucor and Rhizopus. So what is the answer? Third one, right? Chytridium and Rhizopus. Third answer. Then question number 11. Which of the following can be found in both birds and reptiles? Constant body temperature only reptiles, ne? Birds lage මොකද වෙන්නේ? Sorry, reptiles, they don't have constant body temperature, right? Birds have constant body temperature. Then four-chambered heart. Birds have four-chambered heart. Keratinized scales. Both have keratinized scales. Lay eggs and incubate. Birds do that, right? Tail consists of vertebral bones. Both. So we can take third answer as well as fifth answer. Question number 12. Correct statement regarding ground tissue system of plants. All are functional living cells at maturity. That is incorrect because why? Sclerenchyma cells, they are dead at functional maturity. Then they are restricted to only cortex region. No. Now, when we take ground tissues, they fill the gap between the dermal tissue and the vascular tissue. Right? Unit 4 at the beginning of that lesson, we learn about these different tissue types. Right? In the ground tissue, they found they fill the gap between the dermal tissue and the vascular tissue. Right? Mainly consist of cortex, outer to vascular tissue, and pith, which is inner to vascular tissue. Right? Next one. Okay. So we are in 12th question. Third one. This tissue system consists of only primary cell wall. No. Sclerenchyma, they have secondary cell walls thickened by large amount of lignin. So the third statement is also incorrect. Right? Then they are undifferentiated groups of cells. Right? Ground tissues, they are differentiated group of cells. So for the 12th one, the answer should be fifth answer. They consist of specialized cells which can be used in short distance transport. Answer should be fifth answer. 13th one, which makes lactic acid fermentation differ from ethyl alcohol fermentation. Now, fermentation, it comes under anaerobic respiration. Anaerobic and respiration that takes place in the absence of oxygen. Oxygen නැති තැන තමයි මේක වෙන්නේ. හරිද? අපි respiration වල ඉගෙන ගන්නනේ there are two types, aerobic and anaerobic. Aerobic takes place in the presence of oxygen, whereas anaerobic takes place in the absence of oxygen. ඊට පස්සේ මේ anaerobic respiration here, fermentation also comes under anaerobic respiration. There are two types, ethyl alcohol fermentation and lactic acid fermentation. Now, ethyl alcohol fermentation, what is the first step? Right? This is same as the aerobic respiration. The first step is glycolysis. Right? Now here, glucose molecule, it is converted into two molecules of pyruvate. Right? Pyruvate දෙකකට කැඩෙනවා. ඊළඟට මෙතනදී ATP දෙකක් release කරනවා. And also it give two molecules of NADH. Right? මේ pyruvate ඊළඟට steps, it involves in two steps. Steps දෙකකට involve වෙනවා. පළවෙනි step එක තමයි this pyruvate, it is converted into acetaldehyde. Pyruvate molecules දෙක acetaldehyde දෙකකට convert වෙනවා. මෙහෙම වෙනකොට මෙතන මොකක්ද වෙන්නේ? Decarboxylation reaction takes place. Decarboxylation කියන්නේ carbon dioxide release කරනවා. Carbon release කරනවා. ඉතින් decarboxylation කියනවා. Right? So this release two carbon dioxide molecules. Then two acetaldehydes converts into two ethanol. Ethanol දෙකකට reduced වෙනවා. Right? And while using NADH, right? Using NADH. අර හැදිච්ච NADH දෙක ඊළඟට මොකද වෙන්නේ? NAD plus වලට convert වෙනවා. හරිද? දැන් බලන්න එතනදී මොකද උනේ? Pyruvates undergoing complete oxidation, correct? Final electron acceptor is a C31. Yes. Then net ATP yield is 2. Decarboxylation occurs. Yeah. Right. බලමු අපි ඊට පස්සේ lactic acid fermentation. Lactic acid fermentation වලදී glycolysis is the first step, right? Here also the first step here glucose converting to 2 pyruvate. Pyruvate දෙකකට convert වෙනවා ATP molecules දෙකක් produce කරන ගමන්. Right? Then pyruvate converts into lactic acid, but no release of carbon dioxide. Carbon dioxide release කරන්නේ නැහැ. So the difference is how this lactic acid fermentation differs from ethyl alcohol fermentation is in ethyl alcohol fermentation, decarboxylation takes place, but no such decarboxylation takes place in lactic acid fermentation. So the answer is fourth answer. Next one, question number 14. The factor which affect the rate of transpiration in minimum amount. Right? අපි දන්නවා transpiration ගත්තහම higher the temperature, greater the rate of transpiration. රස්නේ වැඩි වෙන transpiration rate එකත් වැඩි. හරිද? ඊට පස්සේ next one, light intensity. Higher the light intensity, greater the rate of transpiration. Third one, humidity. Increase in humidity now. When the humidity outside the leaf surface is low, rate of transpiration is high. Right? Then wind speed, right? Windy conditions, they increase the rate of transpiration. Last one, increase the amount of usable soil water. Soil water, when the soil water amount is high, rate of transpiration is high. So the third statement is incorrect. Humidity වැඩි වෙනකොට නෙමෙයි, humidity අඩු වෙනකොට තමයි transpiration rate එක වැඩි. Right? Answer is third answer. Question number 15. The responses which initiate by blue light photoreceptors are selectively. අපේ මොනවද photoreceptors? There are two, blue light photoreceptors and phytochromes. Then blue light photoreceptors, they initiate a variety of responses, right? There are three: phototropism, light-induced opening of stomata, light-induced slowing of hypocotype elongation. ඊළඟට අපි phytochromes ගත්තාම they absorb mostly red light. Phytochromes, right? They regulate seed germination and shade avoidance. So for the 15th one, answer should be second answer. Next one, question number 16. This question is related to the following, right? Give me a second. Very slow. Right. Then this is regarding phloem translocation, right? මෙතන අහන්නේ question number 60. මෙතන අහනවා අපේ What is the correct sequence of phloem translocation? Plan A. Unloading of sugar in sink. B. Phloem sap moves from source to sink by bulk flow. Then generates a negative pressure in the sieve tube due to the take up water from sieve tube to xylem. Take up water from the xylem by osmosis generate positive pressure in the sieve tube. Last one. The water potential is reduced in the sieve tube by unloading of sugar. Then what is the hypothesis used to explain this pressure flow? Sorry. What is the hypothesis used to explain this phloem translocation? Huh? What is the hypothesis? Send your answers through the chat. What is the hypothesis used to explain phloem translocation? What is phloem translocation? Phloem translocation කියන්නේ මොකක්ද? Transport of products of photosynthesis through phloem tissue. It is called phloem translocation. What is the hypothesis used to explain this? වැරදුනට කමන්නේ answers එවන්න. මතක නැහැ. Pressure flow hypothesis, right? Pressure flow hypothesis. Here මොකද වෙන්නේ? දැන් ඉස්සෙල්ලාම sieve tubes වලට right මේ sugars ටික load කරනවා. හරිද? sugars ටික sieve tubes වලට load කරද්දි මොකද වෙන්නේ? මොකද වෙන්නේ? ඒගොල්ලන්ගේ ඇතුලේ තියෙන දැන් solutes වැඩි. water potential එක reduce වෙනවා නේද? right? source එක ගාව තියෙන sieve tube elements වල water potential decreases. Then, as a result, sieve tubes, they take up water from the xylem by osmosis. ළඟ තියෙන xylem ගෙන් මොකද කරන්නේ? වතුර ඉල්ල ගන්නවා. කොහොමද osmosis වලින්? right? මෙහෙම වතුර ඉල්ලද්දි මොකද වෙන්නේ? It generates a positive pressure. right? Positive pressure එකක් ඇති කරනවා. Force එකක්. It forces this sap to flow along the tube. right? Then ඔන්න sugars ටික අවශ්‍ය තැනට ගිහිලා sink එකට අරන් ළඟට ගියාම මොකද කරන්නේ? අර sieve tube element එකෙන් ඒ ටික unload කරනවා. unload කරාදී මොකද වෙන්නේ? ඔන්න දැන් ආපහු solute potential අඩු වෙලා water potential එක එතන වැඩි වෙනවා. water potential දැන් sieve tube element එකේ වැඩි. ඒ හින්දා මොකද වෙන්නේ? ආපහු phloem එකේ ඉඳන් xylem එකට අර වතුර ටික අයින් වෙනවා. හරිද? right. ඔන්න ඔය ටික කොහෙද explain කරලා තියෙන්නේ? unloading of sugar නෑ unload කරන්න කලින් ඉස්සෙල්ලාම load කරන්න එපා. 16th one. Unloading of sugar in the sink. Right. Yeah, that is correct. Then ඊළඟට අපිට ගන්න පුළුවන් C නේද? Generates a negative pressure in the sieve tube due to take up of water from the sieve tube xylem. Right? ඊළඟට අපිට ගන්න පුළුවන් B. Phloem sap moves from source to sink by bulk flow. No. What is the correct order? Yes, Vidushini, correct your pressure flow hypothesis. The water potential is reduced in the sieve tube by unloading of sugar. Take up water from the xylem by osmosis, generate a positive pressure. හරිද? ඉස්ස වතුර ගත්තන් පස්සේ මොකද වෙන්නේ? ඔන්න positive pressure එකක් ඇති වෙනවා. Phloem sap move from source to sink by bulk flow. Generates a negative pressure එන්න ඕනි C එන්න ඕනි පහළට. අයින් කරද්දී වතුර අයින් කරද්දී තමයි එතන ඒක එන්න ඕනි. xylem එකට වතුර අයින් කරද්දී. right? එහෙනම් හරි උත්තරේ the water potential is reduced in the sieve tube by unloading of sugar. right? Generates a negative, then phloem sap moves from source to sink by bulk flow. මෙතන B වලින් එකක් නැහැ. ඒ හින්දා අපි ගමු ඉස්සෙල්ලාම E, right? B, B, A, C. Fourth answer. Right? Fourth answer. Structures that is found in bulk feeders for feeding. Right? මම Google form එකේ මාක් කරලා තිබ්බ answer එක වැරදි. එතනත් වැරදිලා තියෙනවා. 13 වෙනි එකෙත් මම හිතන්නේ 5 එන්න ඕනේ ඩිස්කස් කරද්දි මම 5 ගත්තේ. මම දැම්මේ 4ද කොහෙද? එතකොට ඒ තැන්වලදී ඔයගලන්ට මාක්ස් එනවා. Structures that is found in bulk feeders. අපේ feeding mechanisms 4යි. Filter feeders, fluid feeders, substrate feeders and bulk feeders. Right? එතකොට animals ලා ගත්තාම දැන් ඔය ඔක්කොම mammals ලා ඔක්කොම මොකද? Bulk feeders ලා. Right? So the structure that is found in bulk feeders for feeding. What we have? මොනවද තියෙන්නේ? මොනවද දත් තියෙනවා? නේ? එතකොට මෙතනදී අපිට ගන්න පුළුවන් tentacles. Tentacles. Fourth one. Select the incorrect statement in relation to connective tissues. These connect organs and tissues structurally and functionally. That is correct. Right? These are fibers. There are fibers and cells in a large extracellular matrix. Correct. Blood tissue is considered as a specific connective tissue. Oh, hey. Matrix is a liquid. Right? Then fibers, they appear only during blood clotting. So this is a specific connective tissue, considered as a specific type. Provide elasticity by elastic fibers in the tissue and reticulate fibers give flexibility and strength. Right? Collagen fibers provide strength and flexibility. Reticulate fibers, they join connective tissues to adjacent tissue. Right? එතන reticulate fiber ගේ function එක වැරදි හින්දා තමයි fourth statement එක incorrect වෙන්නේ. So the answer should be fourth one. Fifth one, heparin and histamine are secreted by the mast cells in the matrix. Right? You know, there are different types of cells in the matrix. So mast cells, they secrete heparin and histamine. So for the 18th question, answer should be fourth answer. Then question number 19. Which of the following is not an enzyme present in pancreatic juice? Right? Trypsinogen විදිහට තියෙන්නේ. Chymotrypsinogen විදිහට තියෙනවා. Carboxypeptidases, yes. Nucleus present. But these aminopeptidase, they are secreted by the intestinal wall. It is not included in the pancreatic juice. So the answer should be third answer. Next one, the element which contribute in acid-base water balance and act as cofactors, enzyme cofactors. Now, you know, our tables ටික පාඩම් කරලා තියෙනවා නම් දන්නවා potassium, sodium, they involve in acid-base and water balance. While magnesium and iron, they act as enzyme cofactors. Enzyme cofactors විදියට ඒගොල්ලෝ වැඩ කරනවා. හරිද? So the answer should be second answer. Question number 15. Then ඔයගොල්ලන්ට තව මාස 11ක් විතර තියෙනවානේ. එතකොට ඔය තියෙන tables, hormone tables, vitamin, mineral tables. හරිද? ඒ ටික මේ කාලේ දවසට අඩුම තරමේ දෙක දෙකටවත් පාඩම් කරන්න. right? නැතුව මේ exam එකට සතියක් තියාගෙන ඕවා පාඩම් කරන්න බෑ. දන්න ටිකත් අමතක වෙන එකයි ඒ කාලෙට වෙන්නේ. right? Which of the following is not a cause for hypertension? Hypertension means sustained elevation of blood pressure above the normal limit. Right? Causes, not a cause. එතකොට causes වෙන්නේ මොනවද බලමු? Obesity, excessive salt consumption, stress, high intake of alcohol, sedentary life cycle, diabetes mellitus. So those are the causes. Cause එකක් නොවන්නේ මොකද්ද? excessive bleeding. ඕනට වඩා bleed වුණාම මොකද වෙන්නේ? එතනදී hypertension වලට එන්න පුළුවන්. හරි? hypo කියන්නේ sustained decreasing blood pressure below the normal limits. Then 22, select the correct pair. මේ මොනවද මේ තියෙන්නේ? Respiratory surfaces, right? Body surface එක පාවිච්චි කරන්නේ කවුද? Cnidarians, flatworms and earthworms. මේ simple animals, right? ඊළඟට gills. Gills තියෙනවා marine annelids, fish, shrimps and prawns. Tracheal system එකක් තියෙනවා insects ලට. ඊළඟට අපි ඉගෙන ගන්නවා lungs. Lungs found in mammals, reptiles, birds, right? Skin එක use කරන්නේ amphibians. ඒ කියන්නේ අර ගෙම්බාගේ අර නිකන් තෙත moist skin එකක් තියෙන්නේ. Right? Book lungs, leaf lungs, scorpions and shrimp. So the spiders and scorpion, not shrimp. Spiders and scorpions. So the answer should be fourth answer. Body surface, cnidarians and earthworm. Question number 23, that is from immunity lesson. Immunity පාඩමින් තමයි ඒක දීලා තියෙන්නේ. හරිද? Select the incorrect response in regard with an allergy. Allergy එකක් හැදෙනවා. Right? Incorrect. Antigens that induce hypersensitive reactions in some individuals are called allergens. That is correct. Right? ඒක හරි. Mast cells are stimulated against allergens and secrete histamine. That is also correct. දැන් allergy එකක් කියන්නේ මොකද්ද? allergy එකක් කියන්නේ කවුරු හරි දැන් හැමෝටම හැමදේම allergy වෙන්නේ නැහැ. සමහරුන්ට ඔන්න අන්නසියප allergy, seafood allergy. ඔය විදිහට එක එක අයට එක එක කෑම, ඊට පස්සේ pollen grains, dust එක ඔය ඔක්කොම allergic වෙනවා. හරිද? ඒ කියන්නේ ඒ person යාගේ immunity system එක මොකද වෙන්නේ? They become overreactive for those particular substances. Right? අනිත් අයට tolerate වෙන දේවල් ඒගොල්ලෝ overreactive වෙනවා. හරිද? ඊට තමයි මේ allergic conditions හැදෙන්නේ. Right? So these antigens, they induce these hypersensitive reactions in some persons, and these are called allergies or allergens. මේ antigens වලට අපි කියනවා allergens කියලා. හරිද? එතකොට allergy කියන්නේ මොකද්ද? Exaggerated response of the body for those certain antigens. එහෙම නැත්නම් allergens. Allergens වලට අපේ body එකෙන් දෙන exaggerated response එකට තමයි අපි කියන්නේ allergy කියලා. එතකොට මොනවද allergens විදිහට එන්නේ? Pollen, right? Dust එක, right? Seafood, pineapple, නේ? ඔවා තමයි allergic වෙන්නේ. ඊට පස්සේ antibiotics, penicillin වගේ, right? ඊට පස්සේ මීමැස්සන්ගේ විෂ, right? බඹරු වගේ විද්දාම ඒ විෂ, right? Those are the allergens. මේ allergic reaction එකක් ඇඟ ඇතුලේ වෙද්දි මොකද වෙන්නේ? tissue injury එකක් වෙනවා. හරිද? These allergens, they stimulate the production of plasma cells. Plasma cells, they secrete antibodies, right? Which are specific for that, for the antigen. When the same allergen enter the body later, right? It become attached to the antibody specific to the allergen and induce the mast cells to release histamine and other inflammatory chemicals, right? So they act on variety of cell types and signals. They bring signals. So the allergic symptoms, sneezing, runny nose, teary eyes, right? Then smooth muscle contraction in airways. එතකොට මොකද වෙන්නේ? හුස්ම ගන්න අමාරුයි. හරිද? ඕක ඕනාවට ඩවුනොත් මොකද වෙන්නේ? අන්තිමටම මැරෙන්න උනා පුළුවන්. හුස්ම හිර වෙලා මැරෙන්න පුළුවන්. Right? Due to breathing difficulty and low blood pressure. හරි. අපි බලමු දැන් මොනවද allergy. So the first statement is correct. දෙවැනියට මම මොකද්ද මෙතන කියලා තියෙනවා? mast cells stimulate against allergens secrete histamine. ඔව් මම කිව්වා. histamine උයි තව inflammatory chemicals release කරනවා. Smooth muscle contraction that carry air to lungs in allergic reaction cause difficulty in breathing. Oh, right? Symptoms are smooth muscle contractions in the airways of the lungs. They can result breathing difficulties. Some of the allergens are pollen, dust, some foods, and antibiotics such as penicillin. That is also correct. When an allergen is entered the body, cytotoxic T cells and antibodies are produced against it. That is incorrect. Ne? මොනවද මං කිව්වේ? Plasma cells තමයි produce වෙන්නේ. Plasma cells secrete antibodies, not cytotoxic T cells. That is incorrect. Plasma cells. එතන plasma cells වෙන්නේ. ඒ හින්දා the answer should be fifth answer. Then question number 24. Which of the following vitamin deficiency causes deficiency symptoms for anemia? Anemia හැදෙන vitamin deficiencies. Vitamins කිහිපයක් තියෙනවා මේ anemia condition එකට හේතු වෙන. දැන් මෙතන vitamins වල නම තමයි දීලා තියෙන්නේ. නැතුව vitamin A, B එහෙම නෙමෙයි නම. ඒ කියන්නේ ඔයගොල්ලෝ name එක පාඩම් කරගන්න වෙනවා. හරිද? right අපි බලමු. thiamine pyridoxine, right? කොහෙද අපිට තිබ්බේවා? thiamine කියන්නේ මොකක්ද? pyridoxine කියන්නේ මොකක්ද? Thiamine means vitamin B1, right? Pyridoxine කියන්නේ vitamin B6. මේ anemia conditions එන්නේ B6 pyridoxine, then B9 folic acid, B12 cobalamin. එතකොට මතක තියාගන්න පුළුවන් 6 9 12 B6 B9 B12 ඒවා අඩු වුණාම තමයි anemia condition එකක් වෙන්නේ. minerals ගත්තාම iron, right? යකඩ අඩු වුණාම යකඩ උණතාවය නිසා මොකද වෙන්නේ? anemia හැදෙනවා. මොනවද එහෙනම් vitamins ටික? B6 B9 B12. B6 කියන්නේ pyridoxine, B9 folic acid, B12 cobalamin. So the answer should be second answer. Then question number 25. Which of the following is correct regarding the process of blood clotting? Blood clotting අපි ඉගෙන ගත්තේ කොහෙද? blood circulatory system එකේ. blood clotting එකක් වෙන එකේ වැදගත්කම මොකද්ද? එතකොට දැන් අපිට සාමාන්‍යයෙන් තුවාලයක් වුණාම මොකද වෙන්නේ? ලේ ගලනවා. එක දිගට ලේ ගලුවොත් මොකද වෙන්නේ? ඇඟේ පුරාම තියෙන ලේ ටික ගිහිල්ලා එතන low blood pressure එකක් ඒ වගේ condition එකක් ඇති වෙලා මැරෙන්න උනත් පුළුවන්. හරිද? ඔන්න ඕක prevent වෙන එක තමයි මේ blood clotting එකකින් වෙන්නේ. blood clot එකක් හැදිලා එළියට ලේ යන එක නවතිනවා. හරිද? right. බලමු. Platelets, vitamin K, and magnesium 2 plus ions act as the factors for blood clotting, right? Platelets, vitamin K, calcium, right? Then damaged platelets release prothrombin, which is a clotting factor. Prothrombin, which is a plasma protein, is converted to thrombin by heparin. හරිද? Next one අපිට දීලා තියෙනවා fibrinogen aggregates and form a network of clot. When the blood vessel is damaged, the connective tissue is exposed and platelets adhere to the collagen fibers in the connective tissue. Right. බලමු අපි මතක් කරමු platelets, blood clotting. Now, when a tissue gets damaged, right, blood flows from it and coagulates and form the blood clot. So this blood clot, it prevents further blood loss and entry of microorganisms or other dust or like other pathogenic microorganisms into the body through those wounds, right? Then normally in undamaged blood vessels, there is no blood clotting takes place. Blood vessel එකට damage එකක් වෙලා නැත්තම් එතන blood clot එකක් හැදෙන්න විදියක් නැහැ. එතකොට අපි කියනවා a highly complex series of reaction, right? That is important. Highly complex series of reaction takes place for coagulation. Blood coagulation එකක් වෙන්න මොකද්ද වෙන්නේ? Highly complex series of reaction takes place, right? And it prevents unwanted blood clotting. හරිද? අනවශ්‍ය blood clotting වීමක් blood clot වීම නවත්තනවා ඒකෙන්. So when a blood vessel is damaged, right, the connective tissues of the vessel wall is exposed. So platelets in the blood which adheres to the collagen fibers in the connective tissue release substances that makes these platelets sticky, right? They release substances and make nearby platelets sticky. හරිද? එතකොට මේ හැදිච්ච platelet plug එක මොකද කරන්නේ? It provide instant protection against blood clots and entry of those pathogenic microorganisms into our body. හරිද? Then platelets, they release clotting factors and trigger the formation of thrombin. Then thrombin converts fibrinogen to fibrin. Next, fibrin aggregates into threads and form a network of clot. Thrombin මොකද කරේ? fibrinogen fibrin වලට convert කරා. හරිද? ඊට පස්සේ මේ activated thrombin ඒගොල්ලෝ තව thrombin හදනවා. And it completes the formation of the blood clot. Right? Then අපි ගත්තොත් heparin වගේම. Right? Heparin, it prevents blood clotting. Right? Heparin prevents the conversion of prothrombin to thrombin and fibrinogen to fibrin. fibrinogen ව fibrin වලට convert කරන්නේ thrombin. එතකොට prothrombin thrombin වලට හැරෙන එක නැවැත්තුවාම there is no conversion of fibrinogen to fibrin. So heparin is used clinically as an anticoagulant. Right? හරි. When a blood vessel is damaged, the connective tissue is exposed and platelets adhere to the collagen fibers in the connective tissue. ඒකේ කොටසක් අඩුයි. Fifth statement එකේ කොටසක් අඩුයි. Fibrinogen aggregates and form a network of clot. That is correct. Right? එතන fibrinogen නෙමෙයි fibrin. ඒ හින්දා ඒකත් වැරදි. Prothrombin, which is a plasma protein, is converted to thrombin by heparin. No, heparin prevents conversion of prothrombin to thrombin. Damaged platelets release prothrombin, which is a clotting factor. එතකොට මෙතන අහන්නේ correct statement එක. මෙතන fifth statement එකේ කැලලක් අඩුවෙන් දීලා තියෙන්නේ. එතකොට එතන marks දෙන්න වෙනවා. හරිද? එහෙම වුණොත් අපි එකට all දෙමු. right? එතන කොටසක් අඩුයි fifth statement එකේ. 26th. right? එතන statement questions තියෙන්නේ. The true statement or statements above the compounds contained in the organism is or are. A. Some storage polysaccharides are formed by the polymerization of monosaccharides belonging to the aldoses group. Right? That is correct. A statement is correct. Then, respiratory pigments of all vertebrates are proteins that have quaternary structure. Right? B statement, that is also correct. C. Triglycerides may have double bonds. Correct. Oxygen hydrogen atoms contributes to the formation of hydrogen bonds in the secondary structure of proteins. Correct. Spider silk fibers are formed by twisting and bending of two polypeptide chains. That is incorrect. A, B, C, D correct. E statement is incorrect. So the answer should be fifth answer. හරිද? ඇයි දැන් protein structure එක ගත්තාම there are 4 levels. In the spider silk fibers, they have secondary structure. එතරම්දි මොකද වෙන්නේ? Single polypeptide chain coils and fold as a result of intramolecular hydrogen bonds and form the secondary structure. It has a secondary polypeptide chain. It bends and fold extensively and forms the tertiary structure. එතකොට ওই bend කතාව තියෙන්නේ මොකේද? tertiary structure එකේ. හරිද? නැතුව secondary structure එකේ නෙමෙයි. ඒකට spider silk fiber කියන්නේ secondary structure තියෙන කෙනෙක් හින්දා ඒක වැරදි. What can be accepted about the evolution of biodiversity? The first eukaryotic fossil is about 3.5 billion years old. First eukaryotic organism estimated as from 1.8 billion years ago. A is incorrect. Then sponges evolved about 670 million years ago. No, 700 million years ago. That is also incorrect. C, fossils of the first photosynthetic organism formed 2.7 billion years ago. Correct. Fossils of the oldest protista about 1.2 billion years old. Correct. First four-legged organisms evolved 265 million years ago. That is incorrect. Should be 365, right? Earliest tetrapods evolved 365 million years ago. So only C and D correct. Answer number 4. Question number 28. Correct comparison or comparisons regarding specific character. Fire. Flame bulb, platyhelminthes, correct. Then, metameric segmentation, nematoda, incorrect. Jointed appendages, arthropoda, correct. Tube feed, echinodermata, correct. Cilia, chordata, incorrect. Right? Answer number 2. Question number 29. Characters distinguish a cartilaginous fish from a bony fish. We can take gills with operculum, right? Then swimming bladder. Then heteroceral tail, right? ඒ තුන තමයි අපිට වෙන් කරලා අඳුන ගන්න පාවිච්චි කරන්න පුළුවන්. A, B, D, right? So the answer should be first answer. Last one, select the correct pair or pairs for the respiratory pigments found in the animal kingdom. Right? Second, ඒකට උත්තරේ එන්න ඕනේ second one. Second one is the answer. බලමු. Respiratory pigments. ඒකත් අපි circulatory system එකේදී තමයි ඉගෙන ගත්තේ. එතන අපි ඉගෙන ගත්ත erythrin, hemoglobin, right? ඊට පස්සේ hemocyanin, chlorochlorine, ඔය විදිහේ ಜಾති ගොඩාරියා ඉගෙන ගත්තා. Right. Hemoglobin එකේ ගත්තාම iron තියෙන්නේ. හරිද? It is hemoglobin, it is present in the blood of human, other vertebrates and annelids. Hemocyanin, it is present in the hemolymph of arthropods and mollusks. Chlorochlorine, it is present in the blood of many annelids. Right? Hemoerythrin, ඒක තියෙන්නේ කාටද? Annelids ලට, some annelids. Myoglobin present in vertebrates. So E incorrect. මතන echinodermata අරගෙන තියෙනවනේ යා invertebrate කෙනෙක්. හරිද? ඊට පස්සේ hemoglobin තියෙන්නේ කාටද? vertebrates ලා, right? Annelids ලා. Human annelida correct. D හරි. ඊට පස්සේ hemocyanin ඒකත් කාගේද තියෙන්නේ? Arthropoda and mollusks. C hari. Chlorochlorine present in the blood of annelids, not mollusk. Right? Hemoerythrin, some annelids. Hemoerythrin found in some annelids. So A, C, D correct. A, C, D correct. Answer number 2. Harida. Right. Now we'll move to essay part. ඔතන ඔයගලගේ MCQ ටික ආපු ගාන 5න් වැඩි කරලා 3ෙන් බෙදන්න. හරිද? එතකොට ලකුණු 50න් කීයද කියලා හොයාගන්න පුළුවන්.